Hello everyone and welcome to today's webinar. The title of today's webinar is Next Generation mRNA Design, Increasing mRNA Potency with a New CAP Analog. Today's webinar is being brought to you by GEN, Genetic Engineering and Biotechnology News, and the CRISPR Journal, both published by Marianne Liebert. And our thanks to our sponsor today, TriLink Biotechnologies. I'm delighted to be the moderator for today's webinar. I'm Kevin Davis, the editorial director for GEN and executive editor of the CRISPR Journal, as well as our new marquee peer-reviewed journal, GEN Biotechnology, which is publishing original research in all aspects of the biotechnology industry. And I'm the author of the book, Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing. Our guest speaker today, we are delighted to be joined by Dr. Chunping Xu, Director of Chemistry R&D at TriLink, where she oversees development of novel CleanCap analogs for improved protein expression and process optimization for modified NTPs. The team is also investigating how to increase mRNA stability using chemical modifications. They've created a library of more than 70 novel CAP analogs to identify next-generation CleanCap analogs. Chunping trained as an organic chemist, completing her PhD at the University of Nebraska, and she's been with TriLink for more than 6 years. Today, we're going to be talking about and reflecting on the interest, the exploding interest in mRNA vaccines and therapeutics, which of course is expanding rapidly, fueled in no short part by the extraordinary success of the COVID-19 vaccine rollout over the last few years. An essential part of the success of any mRNA therapeutic is, of course, the 5' cap structure. And today, Chunping will discuss the latest progress in next-generation mRNA design and reveal how TriLink is continuing to innovate in this technology as the company debuts a novel Cap1 structure. After her presentation, we invite you to stay tuned for our Q&A session. We welcome your questions at any time for Chunping during the webinar. Just follow the prompts on screen. For scheduling reasons, Chunping won't be able to answer your questions live today, but we will forward your questions to her team, and they will do their best to respond to you directly after the webinar. Okay, it's time to get today's presentation underway. Dr. Chunping Xu from TriLink is here to talk about next generation mRNA design. Chunping, thank you so much for being with us. Over to you.
Hi, everyone. Thanks for coming to the GEN webinar today. My name is Chunping Xu, and I'm the Director of Chemistry R&D at TriLink Biotechnologies. Today, I'm very excited to present to you our newest CAP analog, CleanCap M6. Which can significantly improve mRNA potency. This project started from my group, and we developed the chemical process for this CleanCap. And I'm very excited to see it gets launched recently, so customers will get the chance to try in their hands. Here is today's agenda. First, I'm going to talk about the history of capping technology. I will touch base about what is mRNA, what is mRNA capping, what kind of cap analogs have been developed in the market so far, and also what kind of capping methods are available. The second part of the talk, I will introduce our newest cap analog, CleanCap M6. I will talk about the chemical modification, the mRNA capping, and also the in vivo mice study for protein expression. And the last part, I will compare the different costs for different capping technologies, such as ARCA and enzymatic capping, and also, of course, our CleanCap capping. Okay, so as many of you are already familiar with, mRNA can be made through in vitro transcription. So IVT reaction can be done in a tube, a flask, or a bioreactor. It is a DNA template guided T7 polymerase catalyzed RNA synthesis. Here is the list of IVT reagents which include T7 polymerase, DNA template, standard NTP or modified NTP. Cap analogs, if you are using co-transcriptional capping, inorganic pyrophosphatase, kinase inhibitor, and of course, reaction buffer. Once mRNA is made through IVT, a mature mRNA structure should look like this. It contains 5' cap, 5' UTR, a coding sequence, a 3' UTR, and also a poly-A tail. A mRNA without a cap or a tail is not a stable mRNA. It can easily get degraded by exonuclease. Chemical modification can be done to each part of the sequence to increase the stability and reduce the immunogenicity. Historically, a lot of work has been done using modified nucleotide such as pseudo-UTP, N1-methyl pseudo-UTP, and 5-methyl CTP to increase the mRNA stability and also reduce the immunogenicity. And also, the mRNA sequence can be engineered to contain GC-rich motif, a modified 5' or 3' UTR to get a more stable mRNA structure. And recently, my group also started to work on chemical modification on the poly-A tail to increase mRNA stability. For today's talk, I will focus on 5' cap modification. So even though 5' cap is a very small part of the mRNA structure, but it plays a critical role for mRNA function. So natural mRNA cap is involved in nuclear export since mRNA is first made in nucleus, and it needs to be exported to cytoplasm for translation. Also, mRNA cap is involved in mRNA splicing. The synthetic mRNA cap is mainly involved in the mRNA translation initiation by binding to ERF4E. Also, mRNA cap is involved in decapping to modulate the mRNA stability and turnover. Lastly, but not least, mRNA cap is also very crucial for the cell to distinguish between self-mRNA and non-self-mRNA during viral infection. When you hear people talk about cap 0, cap 1, or cap 2, you may be wondering what are the differences? What does that mean? So cap 0 refers to the first nucleotide in the mRNA has 2' OH. So cap 0 is not very abundant in high eukaryotic mRNA. So this is considered not a natural cap. Therefore, it may trigger some innate immune response. One example of cap 0 is ACA. On the other hand, cap 1 refers to the first nucleotide of mRNA has 2' O methyl group. So cap 1 is found in all eukaryotic mRNA. So this is a natural cap and it can promote translation. All the CleanCap from TriLink is cap 1 structure. Cap 2 means there are 2' O methyl on both the first nucleotide and the second nucleotide on the mRNA sequence at the 5' end. So cap 2 are found in 50% of eukaryotic mRNA. So this is considered a semi-natural cap. Cap 2 has not been studied a lot. So the role of cap 2 is not very clear yet. A recent paper from Professor Samuel Jeffries' group shows that cap 2 is a dynamic marker for mRNA agent. So a cap 1 mRNA, if exists in the cell long enough, it can be slowly converted to cap 2 mRNA. Now, since we talked about mRNA and mRNA capping region, now the question is, how did mRNA get capped? So traditionally, there are two general capping methods, enzymatic capping and co-transcriptional capping. I will come back and touch base on enzymatic capping later on. For now, I will focus on co-transcriptional capping. So on the right side, here is a list of all the co-transcriptional capping reagents. MCAP is a very early generation of co-transcriptional capping reagent. People don't use this anymore because there's a lot of issues, and mRNA can extend from the wrong side, N7G side. So ARCA was developed to fix that issue. ARCA directs the mRNA synthesis from the right direction on the right gene. Both MCAP and ARCA delivers cap0 structure. And the CleanCap reagent was developed at TriLink to address the issue of the low capping efficiency from ARCA. And also CleanCap delivers cap1 structure. Now, let's take a closer look at the comparison between ARCA and CleanCap. First, on the structures. We know ARCA is a dinucleotide cap0 structure, and CleanCap is a trinucleotide cap1 structure. Under the same IVT condition, ARCA normally gives around 80% capping efficiency. And due to the initiation competition from the GTP. On the other hand, CleanCap gives about 95% capping efficiency due to the efficient initiation through the dinucleotide AG. And it can easily outcompete the GTP initiation. So in order to improve the ARCA capping efficiency, we have to start with GTP, which will result in low mRNA yield. That is not ideal for mRNA synthesis. And the CleanCap technology, we don't need to limit the NTP, and it normally gives very high capping efficiency and high IVT transcription yield. Let's also take a close look at the comparison of capping process for ARCA and CleanCap. So for ARCA, due to the low capping efficiency, extra reaction, phosphatase, and X purification need to be introduced to remove the 20% uncapped mRNA. So it makes the process longer. CleanCap, since due to the high capping efficiency, we only need one purification. So it's a much more streamlined process. And also CleanCap gives higher transcription yield. So since the development of the first CleanCap at TriLink, our Chemistry R&D group never stops the innovation. We want to further optimize the CleanCap for better activity. We found that by adding an extra 3' methyl to the M7G, it can further improve the protein translation. So here is a table shows the mRNA quality attributes. So this is the luciferase mRNA that's being capped with ARCA and the two different CleanCap from TriLinks. And we compared the IVT reaction yield, integrity, capping efficiency, double-strand RNA level, residual DNA, and the residual protein. From this table, it's very clear that both CleanCap AG gives better IVT reaction yield, better integrity, and also better capping efficiency, and a low double-strand RNA level, which is an undesired critical mRNA impurity. Regarding to the residual DNA and residual protein, all these three caps are very comparable. Now, once we have the capped mRNA, in order to do the in vivo study, we need to formulate the mRNA with LNP and then inject to mice. Historically, TriLink has been using luciferase to monitor the protein expression. So luciferase was capped with ACA, CleanCap AG, CleanCap AG 3' O-methyl, and then formulated the same way with LNP and injected into mice for in vivo protein expression. And this is the image of the in vivo luciferase expression. So the luminescence expression level was monitored at different time points from 3 hours to 24 hours. And from this image, you can clearly see the trend, the increase in protein expression from ACA to CleanCap AG to CleanCap AG 3' O-methyl at 3 hours, 6 hours, 9 hours, and 12 hours. And this graph is an actual quantification for protein expression. And the trend is very clear. The extra 3' O-methyl on the same N7G has big impact on the protein expression. So this work we also captured in our white paper. You can find it at TriLink website. During COVID pandemic, if you get the Pfizer-BioNTech vaccine, if you ever wonder what caps have been used in that vaccine, I'm very proud to tell you that they used TriLink's CleanCap AG 3' O-methyl. And we are very glad that we can contribute to the vaccine development with our CleanCap capping technology. Now I'm going to move to the second part of the talk. I'm going to introduce to you our newest cap analog, CleanCap M6. TriLink Chemistry R&D team has been actively working on modifying and optimizing CleanCap for better activity. So the modification is focused on N7G, the triphosphate linker, and also the first nucleotide A. A library of more than 65 unique cap analogs have been synthesized and tested. So most of these cap analogs have been incorporated into luciferase mRNA, and we evaluate capping efficiency, the IVT yield, the mRNA integrity, and also double-strand RNA level, residual DNA, temperate, and the protein level. And more than 20 of those cap analogs have been tested in the in vivo mice study for protein expression. Out of all these screening efforts, I'm very proud and excited to announce that we discovered and launched our new cap analog. Here is the structure of CleanCap M6. And you may be wondering, what is the new modification for this molecule? And it is the N6A methylation. So even though it's just adding an extra methyl group, but that all it matters for protein expression. And also this methylation modification is not a random design. It's based on some literature report. We know the N6 methylation happens in mRNA in nature. And also some literature paper reports that the M6A methylation will help to control mRNA stability by slowing down the decapping process. Here is the structure overview for different modification and function. So the charged N7G is required for EIF4E binding. And we know the 3' O-methyl can enhance protein expression. The mechanism is not clear yet, but we see this pattern across different modified CleanCap. So the impact is real. And the 2' O-methyl on the A confers a cap1 structure, which gives a natural cap and can promote translation. And the N6A modification, which is new, and what we believe is It can stabilize mRNA by slowing down the decapping process. Here is a side-by-side structure comparison for 3 different CleanCap from TriLink. So this is on the left, it's the very early first generation of CleanCap developed at TriLink. And the middle one is our second version, CleanCap AG 3'O methyl. Also, this is the one used in the Pfizer COVID vaccine. And on the right side, this is our newest CleanCap M6. So all the 3 CleanCap, they are all Cap 1 because they all have the 2'O methyl on the A. So the CleanCap AG 3'O methyl has actual 3'O methyl on the N7G. And the newest cap have both 3'O methyl and also N6 methyl on the A. All the 3 CleanCap analogs, they give very high capping efficiency due to the efficient initiation through dinucleotide AG. And the CleanCap M6 AG was incorporated into different mRNA constructs, such as EGFP and F-loop and Cas9. They have very different lengths from 1 kb to 4.5 kb. And the mRNA quality attributes are compared in this table here. And we are very glad to find that mRNA capped with CleanCap M6, they all maintain high IVT reaction yield, high capping efficiency, and reasonably low double-strand RNA level. And also this 3 CleanCap from TriLink, we also compared their in vivo protein expression in mice. So the 3 cap analogs, they were incorporated into luciferase mRNA, formulated with LNP and inject to mice. And this image is from 24-hour post-injection. And we can see the clear trend, the protein expression enhancement from CleanCap AG to CleanCap AG 3'O methyl to CleanCap M6. And this is the actual quantification. And each modification marks a significant increase in the protein expression level. Further, in vivo dose study shows that since CleanCap M6 can offer a more potent mRNA, that means we can use lower dose instead of regular standard dose 1 mg per kg. We can use 0.3 mg per kg of mRNA to achieve the same protein expression level, which is a good news. Less low dose means less immune response. Up to this point, we know the advantages of CleanCap M6 AG. And one thing I haven't convinced you yet is how about compared to the classic enzymatic capping? So we did do side-by-side comparison. So luciferase mRNA was capped with both enzymatic capping and co-transcriptional capping, are formulated in the same way with LNP and inject to mice for protein expression study and comparison. Here are some mRNA quality attributes for luciferase mRNA capped with either enzymatic cap or 3 classic TriLink CleanCap, including the newest CleanCap M6. And we are very glad to find out that the IVT reaction yield, mRNA integrity, and capping efficiency, they are all comparable to the enzymatic capping. And the CleanCap M6 even offers low double-strand RNA level. Regarding to residual DNA and the residual protein level, all these 4 caps are very similar and comparable. Here is the in vivo study. between mRNA capped through enzymatic and also mRNA capped with CleanCap M6. This is an image of 12-hour post-injection. And we can see the obvious protein, the luminescence enhancement from enzymatic to M6. And the middle graph is a time course from 0 hour up to 48 hours. And clearly we can see the mRNA capped through our CleanCap M6 outperforms the mRNA capped through enzymatic capping at each time point. And this is the actual quantification of luminescence expression level. Now let's also compare the capping process for both enzymatic capping and CleanCap capping. We know enzymatic capping a lot of times means using multiple enzymes and multiple steps. And also enzymatic capping normally happens after post-IVT reaction. So that's extra step. So that makes the process longer. On the other hand, CleanCap, since it's a streamlined one-part IVT reaction, it makes capped mRNA directly. So it's just reaction, purification, and then final mRNA. Both of this capping process offers very high capping efficiency and transcription yield. Up to this point, I hope I have convinced that our CleanCap M6 has these advantages. It can make mRNA express more protein. That means we have the potential to lower the dose of the mRNA drug for clinical use. And also low dose means less immune response. And also CleanCap M6 can maintain the high IVT yield and high capping efficiency from the different mRNA construct we have tested so far. And also our streamlined CleanCap technology process can make the manufacturing process faster. So it will make the turnaround time faster and allows the time and the labor and the cost to manufacture. Now I would like to move to the third part. After you heard about all the scientific part, you may be wondering, how about the cost compared to enzymatic capping or ACA capping? So our product development department, they did a third-party extensive customer survey to address the reagent cost, labor cost, purification cost, total manufacturing time, process development time, and the challenges at large scale manufacturing for different capping technologies. I would like to share some of the survey results in the next few slides. First, reagent cost. So capping process using CleanCap, enzymatic capping, and ACA have been compared. And the costs are very similar. CleanCap may be slightly lower, but not significant. So I should say reagent costs for 1 gram of GMP grade mRNA, all these 3 cap analog are very comparable. Now the process development time, since CleanCap technologies offers a more streamlined process, so it will take less time to optimize the process and implement the process. So from the customer survey, for CleanCap process development time, it takes 2 months. And for ACA and enzymatic capping will take 4 months. So it cut down the process development time by half, which is very significant. And also, how about the total manufacturing cost? The total manufacturing costs include reagent cost, purification cost, and the labor cost. By adding all the together, we can compare the total manufacturing costs across the 3 capping technologies, CleanCap, enzymatic and ARCA. And from this graph, we can see the enzymatic capping will cost 30% more than CleanCap, and ARCA will cost 20% more. So this cost is based on 1 gram of GMP mRNA. And of course, when the scale goes up, it could be a different scenario. And the increase of cost is not linear to the scale. So let's compare at 40 gram GMP mRNA. So it will cost 40% more to do enzymatic capping than CleanCap, and 30% more to do ARCA capping than CleanCap. So it will be very critical for the cost when you move to the late stage clinical trial. So from cost and process-wise, I hope I have convinced you using our CleanCap M6 technology, it can reduce both reagent and labor cost. And also it will cut your process development time by half. And also CleanCap M6 will guarantee you a Cap 1 structure with no extra steps. And for enzymatic capping, it can provide either Cap 0 or Cap 1, depends on the choice of enzyme. So for those of you who haven't heard about TriLink Biotechnologies, I would like to briefly introduce who we are. So TriLink Biotechnologies are not just capping technology company. We also make standard NTP, modified NTP, such as N1-methyl-pseudo UTP, and both at IUO and GMP grade. TriLink also provide custom oligonucleotide synthesis and custom chemistry synthesis. Recently, TriLink also developed the capacity to do plasmid manufacturing for mRNA synthesis. And more importantly, TriLink has been doing mRNA service for more than 25 years. We have served close to 1,000 customers across the world, and more than 100 batches of GMP mRNA grade have been made in our facility. TriLink also offers process development for mRNA synthesis. So basically, TriLink offers service from pDNA to IVT reaction to mRNA and to analytical service, the entire service. And also, to speak to my heart, innovation. We have a very strong R&D team. The innovation never stops. We are working very hard to come up with new product, superior product to go into the market. And these are some pictures of our facility. So TriLink is located in beautiful San Diego, and our headquarters is Water Ridge facility. This is where my office is located. We also have 2 extra GMP facilities to make GMP grade NTP, CleanCap, and also mRNA. And all this work I presented today, it's contributed by different groups, from R&D to PD to manufacturing to product development to QC and to marketing. I'd like to thank every team members I listed here for their contribution to this project. Lastly, I'd like to thank you for your attention. And we do have a Q&A questions after this talk. Thank you.
Thank you, Chunping, for that excellent presentation and showing the wonderful work of your team at TriLink, working on next generation RNA design. So Chunping, first of all, can you share any details on why the M6 analog has so much higher protein expression?
Good question, Kevin. Yeah, so we believe, that M6 methylation can slow down the mRNA decapping process, and it will improve the mRNA stability. So which contributes to better potency in the in vivo protein expression. And there is a literature support for that.
Okay, great. Thank you. Have you done any work on increasing in vitro transcription yield?
Yes, we actually have done extensive IVT optimization. And, the yield I reported in this presentation is around 4 to 5 mg per ml. But, our biology team, we did optimization, we did a pulse study, so introduce NTP, at a different phase, and which can increase the IVT yield up to 10 mg per ml.
Have you looked at additional genes in your in vivo models?
Yes. So from this presentation, I mainly focused on luciferase mRNA, but we also incorporate, the CleanCap M6 into EPO, and we did an in vivo study as well. We are also in the process of incorporating the CleanCap M6 into mCherry mRNA, and also EGFP, Cas9. So yeah, we tried on different mRNA construct, and it seems to all work very well. Yeah.
Is M6 available in GMP grade?
We are in the working process, and depends on the market need. We are planning to launch GMP grade M6 cap next year, 2024.
Great. What modified NTPs have you tested so far?
Yeah, so we have tested, the classical ones, the N1 methyl pseudo UTP, and we also test the pseudo UTP. We test the 5-methoxy UTP, and we are also in the process to testing more modified NTP. Yeah.
Okay. Just a few more questions before we close, Chunping. I'm not sure when M6 was released on the market. Can you say what the initial response has been from some of your customers?
Oh yeah, that's a very good question. So we launched this product in the TIDES meeting at the beginning of May in San Diego. So actually, right after the meeting, we get a lot of customer interest, even, actually real customer order. So I would say, yeah, it's well received. Yeah.
Well received. It's obviously very early days. So
yeah.
It's a slightly unfair question, but that's good to know.
Yeah.
We entitled this webinar Next Generation, or you entitled the webinar Next Generation mRNA Design. What other areas is your team studying or working on as far as next generation mRNA design goes?
Yeah, I'm glad you asked.
Ones that you can tell us.
Yeah, I'm glad you asked this. So my team, our Chemistry R&D team, recently we started to work on poly-A-tail chemical modification, because we feel there is a great need in this area, and it has not been studied a lot. And the chemical modification of poly-A-tail is equally important as cap modification. So that's we are working on, and probably we are hoping to see the next generation of mRNA from that area.
Wow, that sounds exciting. Well, we'll have to have you back for another webinar to talk about your work and launches in that space. And finally, you did a lovely job of sort of showing the timeline of development and the architecture of these evolving cap structures that have been developed. Is M6 the last word, or are you continuing to investigate additional modifications?
No, definitely not the last, but our chemistry R&D team are still actively working on novel caps, more modification. We actually made a big library. A lot of them have not been tested, and a lot of them looks very promising. So we are looking forward to the even more, like a better version coming out soon. Yeah.
Well, I'm sorry we can't take any further questions right now, but I'm sure if people are interested and have questions for you, they can reach out to you offline or any member of the TriLink team, and I'm sure you'll do your best to help them out and provide
Sure.
some additional clarification.
Yeah,
I knew you would say yes, so that's great.
Yeah.
Thank you so
much. Happy to assist that. Yeah.
Of course. Thank you again, Chunping Xu, Director of Chemistry R&D for TriLink Biotechnologies. That was a really fascinating and important webinar presentation on next generation mRNA design. We want to thank TriLink for not just employing you and helping sponsor this research, but sponsoring today's webinar as well. The webinar was produced by Leanna Jabs and Hannah Turner from Jen's outstanding multimedia group. And so all that remains is for me, on behalf of everyone at Jen and the CRISPR Journal, who've hosted today's webinar, to say thank you. I'm Kevin Davis. Thanks for watching.
Thank you all.
Bye-bye for now.
Bye.